Thank you for that terrific talk. The next speaker is Shawn Davidson. Shawn was a member of the biology department graduate program and conducted his PhD work in the lab of Matt van der Heijden in the Koch Institute. Here he made major contributions to understanding the role of metabolic pathways in cancer development and progression. And I think Matt presented some of his work in this morning's talk about the effect of metabolism in serum. He graduated in 2017, did a short postdoc with Ollie Jonas, who's actually our next speaker after him, and then he moved into an independent position as an associate research scholar and lecturer in the Lewis-Sigler Institute for Integrative Genomics at Princeton University. Here he continues to investigate the interface between genetics and metabolism in normal versus diseased tissue using highly quantitative approaches with the goal of discovering new therapeutic strategies. And the title of his talk is Spatially-resolved Assessment of Metabolism. Please join me in welcoming Shawn.
Great. I'd like to thank the organizers, especially Matt, Tyler, and Jane. And I'll be talking today about spatially resolved assessment of metabolism. So it means a lot to be here today to celebrate 10 years at the Koch Institute. I was an undergraduate doing research in Building 68 while watching the construction of this back in 2010. At that time, I didn't know that I would be so fortunate to be a graduate student here, and even more fortunate to be one of Matt's first PhD students in the Koch, and it was a really wonderful time. I have to say that, consistent with what Matt was saying earlier, that Matt really embraced the vision of the Koch Institute from the beginning. And here's some pictures of some of the things he described earlier. But we were able to miniaturize plasmapheresis for the mouse, which really translated into this optofluidic CTC cell sorter. And then even separate from that, I started working with a Microdevice with Oliver Jonas, working with Bob Langer at the time. And it's hard to imagine being able to do any of those, never mind all of them, in one place. So it was a terrific opportunity. My main focus in Matt's lab at the time was looking at what do cancer cells eat, and what nutrients are essential for tumor initiation, progression, and what nutrients might be at distant sites that might be ideal for tumors to grow. And so we know from this that this was kind of the view of cancer metabolism in 2010. We know the Warburg effect, so cancer cells use a lot of glucose and they make a lot of lactate in the absence of oxygen. But over the last several years, we found that the kind of modern view of cancer metabolism is that there's really as many different phenotypes of cancer metabolism, and as a result, many new drug targets. And tumors use all of these major metabolic pathways in completely different ways. And so the goal of a lot of my PhD work and a lot of my ongoing work is to try to figure out which metabolic vulnerabilities might be there in these settings. So what influences the metabolic vulnerability, metabolic phenotypes of these tumors? Well, this is a nice figure from Matt talking about intrinsic factors. And so some of these, like genetic alteration, different gene mutations, cell of origin, these you could study in cell culture. And so that's a lot of what was being done. But we realized that these extrinsic factors, like the access of these cancer cells to nutrients and oxygen, and other things like microbiome, are just difficult to study in a dish. And so a lot of my PhD work was dedicated to how can we standardize these physiological systems to control for these individual and very complicated variables. And the approach we took was this in vivo infusion of stable isotope tracers in autochthonous mouse models of cancer. And so we would do surgery to get access to the jugular vein and infuse a 13C or 15N or any other stable isotope labeled tracer into the mouse. And then because we know from classic enzymology and biochemistry, the atom transitions from your starting tracer, which in this case is glucose. We can follow each one of these carbons through different pathways, and shown here glycolysis. And we know that 1, 2, 3 carbon here corresponds to 1, 2, 3 carbon here by pyruvate, and we can measure that by mass spec. And so using that approach, we found many differences between kind of these tumors in vivo compared to in vitro, specifically that tumor cells in vivo in this lung cancer model don't use much glutamine, which cells in culture do. And so this was a good start, and we've learned a lot using this technique. But this still relied on bulk measurements, and so as everyone is moving into kind of these spatial analyses, I'd like to talk today about the work that we've done at Princeton to get metabolism into the age of spatial omics. So how can we get past these bulk metabolism measurements? Well, this is kind of what we would conventionally do. We would take a tissue, we would grind it up, and we would run it on LC-MS. The input of cells would be 10 to the 5th cells required for this assay. So you can't really get to the single cell level. And then if you want to study a specific population of cells, you might be able to use FACS, but a lot of things changed during the sorting process. So we needed to do something different. So the challenges associated with making these measurements are that, in contrast to something like transcriptomics, is that there's no amplification. You can't do a PCR for pyruvate. And there's just a wide concentration range with a high degree of chemical complexity. And so the approach that, So if we could do this spatially, then we would be able to address important questions. And so we know that there's a lot of diversity in normal tissues and cancer, and shown here is GBM and PDAC, which we have ongoing projects. And if we could do this, we could start to study these processes over the course of development and disease progression, and specifically questions that could be tested or generate other hypotheses are those about cell interaction, those about cell organization, how nutrient gradients play roles in organization during development, for example, how cells use nutrients to power fundamental processes, and the last question, which I'm most interested in, is how cells compete for nutrients in the tumor microenvironment. And then the other way to think about these spatial technologies is perturbation setting, where you can do genetic, drug, or infection, and you can start to ask questions about how metabolism changes during disease progression, or in response to therapy, or in response to diet. And so to start to get at these questions, we turned to start using a technique called matrix-assisted laser desorption ionization coupled to imaging mass spectrometry, or MALDI-IMS for short. We have an input of fresh frozen tissues. We can get a spatial resolution of around 5 to 50 micron, depending on the analyte. We can achieve relative and absolute quantification of individual metabolites. We can perform histology on that and start to link these data sets together. And critical to MALDI is which matrix you select. So here's just a couple of matrices that we use in the lab for negative ionization, but you can see that will dictate what kind of molecular coverage you get for that. And then we can also overlay these things with histology-based database. On the left, you can see this is dimensionality reduced for our MALDI data, and this is the Allen Brain Atlas, and we get some nice concordance between the anatomical features. So there are metabolite signatures that kind of correspond with those structures. And this would be a typical slide layout for some of our integrated omics experiments. So we will typically do MALDI in positive, negative, and then we'll do histology and pathology, and then take extra slides for things like slide seq and cell painting. So critical to getting this technology to work was an initial collaboration with Bruker Daltonics, and basically working with them, we were able to kind of shift the distribution of molecules we can see from kind of the higher end where you see a lot of lipids and other things to really getting small molecule metabolites. And we use this Bruker Celerix 9.4 Tesla FTICR. We had to develop some data analysis pipelines, so we wrote a program called Isoscope, which can be used to analyze the resulting data from these MALDI experiments and has some nice features and is usable in open source. And we've also been using some of the other spatial technologies for these, for our more unlabeled pipelines. So with all of those things in place, we were able to start establishing what we've called isoimaging. So today I'll talk a lot about using isoimaging in the context of normal tissues and then show some snapshots of ongoing work and how they apply to cancer. So the first thing we had to do was we needed to do benchmarking to see which metabolites we could see and which ones we could trust to measure. So we compared this to our HPL CMS, and we found 7 quantitation metabolites that had similar labeling patterns in LCMS compared to MALDI. And we've also been able to validate over 240 different metabolites by on-tissue MSMS. So that's our kind of conventional method. And we can start to use isoimaging to assess different things. So first, let's talk about metabolic pathway activity. So if you're interested in probing glycolysis, you could infuse a labeled glucose into the mouse, and then you can follow glucose into UDP glucose. And if you see an M6 labeled glucose, you would know that that has gone through glycolysis. And if you wanted to look at gluconeogenesis, you would infuse a 3 labeled glycerol, and that would, if that goes through gluconeogenesis, you would wind up with a 3 labeled UDP glucose at the end, and so that's what we're monitoring. And here you can see in the medulla that the medulla is more glycolytic. There is more signal here, and in the cortex, you can see that it's more gluconeogenic because there's more M+3 labeling here. So this is starting to probe pathway activity. Next, we can start to measure things like direct contribution and metabolic flux. And so the power of some of these assays, so people have done MALDI for metabolomics for a while now. So with metabolomics, you can get a quantification of how much of a metabolite is there. But with these relative flux and direct contribution assays, you can start to ask questions, how fast and from where? And so I hope to convince you with this next example that this initial measurement of malate being the same in the cortex and the medulla is actually much more interesting than that because of where it comes from. So here's the visual. So you can see malate levels are pretty similar in the cortex and medulla. We've infused 30 different tracers into these animals, and these are 3 that show examples that contribute relatively equally to that malate pool in the kidney. But then if we look at cortex-enriched contribution, we can see something like citrate, which is one of the extreme examples, really only contributes to malate in the cortex. And then similarly, we show that fatty acids contribute more in the medulla. So when you're thinking about things like vulnerabilities and essential nutrients for cancer, it's important to know where they're coming from, because if you target one pathway, there may be another pathway that's making that metabolite. So knowing this is very helpful. And then so we can quantify that, and just kind of, we can account for almost all of the carbon in malate in the TCA cycle. So that covers direct contribution. So we can then also get true spatial metabolic flux analysis by doing these kinetic infusions. So we can do a serial sacrifice of animals and collect time points, and then look at the labeling in the spatial regions. And so we can start to model flux by having these kinetic infusions. And here you can see, the TCA flux is much higher in the cortex, for example. This can be used to revisit some fundamentals in metabolic regulation. And so these are some low throughput examples, and we have some higher throughput examples ongoing. But here's a good example where we can localize our metabolic activity with protein expression or enzyme expression. So here we infuse a 5-labeled glutamine, and if the enzyme glutaminase is present, you would see a 5-labeled glutamate. And so you see, pretty good distribution of 5-labeled glutamine in these kidneys. And then this can be explained by the fact that glutaminase is only expressed in the cortex. And so this tracks nicely because you only see labeled glutamate in the cortex and not really here at all. And that's just because the enzyme is there. So you can imagine doing this in a much higher throughput way and getting all the transcripts and all the expression for different metabolic enzymes. So think we can learn a lot on the fundamental regulatory level. We've used this to start doing dietary perturbation. So here's an example of the brain. And this is what you would learn from your biochemistry textbook. If you ask where does glutamate come from in the brain, you probably get the answer that most of the carbon would come from glucose. And so that's true. It's not very interesting from a spatial perspective. But it's much more interesting when you start adapting these to a ketogenic diet, and then nutrient preference really changes quite dramatically. So when you put animals on the ketogenic diet, they don't use as much glucose to fuel the TCA cycle to make glutamate, and then they start using ketones. And what I think is especially interesting about this part is the rate at which they change and kind of the adaptability of each of these different regions of the brain. And so I think it would be exciting to probe more on the function side of why these regions here seem to switch to using 3-HP in different ways, or faster than other regions of the brain. And in contrast to just infusing glucose and asking where all the carbon for glutamate comes from, we infuse different nitrogen sources to ask where the nitrogen source for glutamate comes from. And the answer here is much more heterogeneous. So you can see in different regions of the brain, they get their nitrogen for glutamate from different sources. So you have branched chain amino acids, so leucine, valine, and isoleucine, and you can see aminoglutamine and ammonium come from different places to make glutamate, which is pretty interesting. And there's a lot of disease implications here. Actually, a lot of these are dysregulated in things like Huntington's and other neurodegenerative diseases, so we're following up on that. So these are kind of just the different ways that you can use this isoimaging approach to get new information. And I'd like to give a few kind of snapshots and future directions on how we're using this to study cancer metabolism and kind of where it's going. And so I think we're getting very close to being able to make these single-cell metabolism measurements. We've been able to improve sensitivity greatly by doing a series of tissue-specific washes. So a lot of the reasons we can't see things is due to ion suppression. And so if we can remove the things like lipids or peptides that are causing ion suppression in certain contexts, we can start to see metabolites at higher, with higher sensitivity, which means when the lasers get smaller and the optics are better, you'll be able to see those metabolites. Because right now we're on the limit of detection for many metabolites. There's a lot of activity in kind of developing new matrices, and these include reactive matrices, which both derivatize and serve as a matrix for accepting a proton. And there's a lot of interest in this and how it can be used for small molecules. And I think with some hardware advances, we'll get even further, so with ongoing collaborations with Bruker. So this spatial metabolomics technique has opened up a number of possibilities to answer questions that we couldn't really access before. So for example, one of the projects we have going is during immunization. So previously it would be difficult to isolate the cells in a germinal center. So we've been able to characterize what nutrients are required for germinal center B-cell metabolism upon immunization with classic agent SRBC. And so we can make these measurements. I think this would be really interesting to start looking at tertiary lymphoid structures. So if anyone has any good models of that and would like to take a similar approach, I'd be highly interested in that. Also looking at things like cancer clonality. So how do cancers with the same genetic drivers perform diverse metabolic programs? So to do this, we've crossed the KPC mice with the confetti allele, and so you get stochastic recombination. So each one of these colors will be a different clone and outgrowth, and then we can start to overlay ion images and figure out there's clear differences in these tumor regions that have active KRAS and P53 loss. We've also started looking at human samples, and this has been a bit challenging, but working with Ernest Frankel's lab here at MIT, we were able to make sense of kind of these 10 patient medulloblastoma study. There's a lot of variables to consider when getting different banked human samples, like collection date and duration of storage and storage conditions. But we went through and did some batch correction and found some metabolites that are very specific to sonic hedgehog A and sonic hedgehog B subtypes of medulloblastoma. In the clinical space, also working with Ollie Jonas, who you'll hear from next, we've been able to use this microdevice and couple it to our MALDI analysis. So the microdevice is implanted into the tissue, and you're looking at a slice of the tissue. The drug has been delivered into this region here, and we can see PEG by using MALDI, and so we can get a good sense of where PEG is, and the drugs are formulated in PEG, and so we can follow the drugs, and then we can follow the response of the resulting metabolic profiles. And as an example, here's drug 1 and drug 2 released in different directions, and you can see the different metabolic profiles that result from kind of the delivery of each of these drugs. And we're overlaying that with different cell type markers, so you can see kind of more T cells down here, more hexo-6-phosphate here, so probably more glycolytic region where the T cells are. And so there's a lot of this, and we're working on the first 6 patients from Ollie's study on that. And then also using the microdevice, we can assess drug response and drug signatures from tumors. So this is an example. You can see it better on the H&E. So here's where the device was, and then the drug is released at 7 o'clock. And you can see this beige region here from dimensionality reduction. And then we can go and pull out the metabolites and ask which metabolites are responsible for the change. So I think we're really close to having robust single-cell methods to do spatial metabolism and spatial flux. And I think a lot of the upcoming integration with other spatial modalities will be really important, and we'll be able to make some exciting discoveries in the near future. These in situ measurements using the microdevice, I think they can be used to identify drug response signatures, and there's a long list of other therapeutic things that we can deliver with those as well that I think Ollie will probably talk about next. So I'd like to thank the people in my lab and all my funding sources, and yeah, happy to chat at some point.